So in this next recording, we're going to address our final group of biomacromolecules, the nucleic acids. So again, we've talked about carbohydrates, we've talked about lipids, we've talked about proteins, and now we're going to talk about nucleic acids. Remember, nucleic acids is one of our true polymers, along with our polysaccharides, as well as our proteins. And the monomer that makes up the polymer that is nucleic acids are called nucleotides. Now, nucleic acids come in two major forms. We talk about RNA and DNA. RNA being ribonucleic acid, DNA being deoxyribonucleic acid. So most people are familiar with the classic double helix of DNA. Not as many people are familiar with RNA, but we're going to address both of these molecules and their functions in much greater detail as we progress through the molecular mechanisms of gene expression later in the quarter. But in this recording, I want to just talk about them in context of biomacromolecules and realize that they are there so we can come back to them later. So again, nucleic acids are polymers. We sometimes call them oligonucleotides, many nucleotides. Again, the monomers are nucleotides. Now, the nucleotide basic structure is something that I need you to be able to draw. And this is a basic outline of what you should be able to draw. And this is going to come into play to have a better understanding of how nucleic acids work. So we're going to lay that foundation down now so that it's easier for you to understand later. But each nucleotide is going to be composed of a nitrogenous base, meaning it has nitrogen in it, a sugar, in this case it's a 5-carbon sugar, and a phosphate group. All right? Now, sometimes you'll hear the term nucleoside, meaning just the nitrogenous base plus the sugar. And this is important because the sugar is going to dictate whether you're talking about DNA or RNA, and the nitrogenous base will then tell you which of the 4 types of nucleotide you're dealing with within those two biomacromolecules. So there are 2 types of nitrogenous bases. We have pyrimidines and purines. So pyrimidines are going to be cytosine and thymine, which we find in DNA, and then uracil, which replaces thymine in RNA. And they have a 6-membered ring. The purines are adenine and guanine, have a 6-membered ring with a 5-member ring attached. So it's going to be a bigger nitrogenous base, shorter word. And that's how I differentiate between the two. So in DNA, the sugar is going to be deoxyribose, again, deoxyribonucleic acid, versus ribose in RNA, ribonucleic acid. And when we look at those bases to reinforce that, we can see now the single 6-membered ring of the pyrimidines and the double ring structure of the purines. Again, uracil we only find in RNA, whereas cytosine, adenine, and guanine are found in both RNA and DNA. Thymine then is only found in DNA. Addressing the sugar component of the nucleotide, we can see here deoxyribose, which we find in DNA, and ribose in RNA. Again, understanding kind of the breakdown of the name of something in biology helps you understand and remember what it means. So ribose is a 5-carbon sugar, and it's in circular form here. Deoxyribose, deoxy means without oxygen. And so as you can see, the 2' carbon has but a hydrogen attached to it, as opposed to in RNA, the 2' carbon is attached to a hydroxyl group below the ring. Right? So without that oxygen, now you have DNA. And that's important to understand because we're going to go on later when we talk about sequencing technologies, we're going to have to introduce the idea of a dideoxyribose. Now that dideoxy is twice without oxygen. And of course then you're thinking, obviously, then the 3' carbon is going to be devoid of a hydroxyl group as well. And that's going to impact how that molecule works. And that's going to be key to the technology of sequencing in its current format. Now, the thing about the bases is that their sequence is how you store information in a nucleic acid. And DNA, being a double-stranded molecule, introduces the idea of complementary base pairing. So when I assemble this double strand, in fact, it's the nitrogenous bases that are interacting, A's with T's, G's with C's, that allow those 2 strands to hold together. Now, the beauty of going back to the theme of form and function is that when Watson and Crick, proposed their model of DNA in this format, it lent itself to a mechanism for replication. if there's this complementary that A's only bind with T's and G's only bind with C's, that if you pull the strands apart, you have now a template to build a new strand. And in the long run then you can obviously build two complete molecules from one molecule. We'll address this in greater detail later, but I want to lay that foundation down now. Again, we call this Watson-Crick base pairing, and it is the key to replicating a piece of DNA. It's also going to be a key to copying DNA to RNA. So that's self-replication and information transmission. Which really brings us then to the heart of molecular biology, which is what's called the dogma of molecular biology. This is gene expression in a nutshell. And again, introducing it today so you can start to think about it, but we'll spend great details, we'll go into great detail in this later in the quarter. But this idea that information is contained in DNA, and that can be copied into an RNA molecule. That RNA molecule can be read to make a protein, and proteins do stuff that result in traits. The color of your eyes, the color of your skin, moving your muscles. All right. So again, here we see DNA can be transcribed, and that is going to be a process of making a copy into RNA, and then the RNA, the information in the RNA, can be translated into a polypeptide. So in essence, the order of the sequences, the sequence of the nucleotides, the order of the nucleotides, codes for individual amino acids, and that's the secret of DNA. Now, in a eukaryotic cell, we can see this process then distributed across the cell. We start in the nucleus where the DNA is maintained. We make a copy of mRNA. That mRNA travels out of the nucleus and is copied by the ribosomes into a polypeptide. Each of these components we will address as we move into cellular anatomy, but again, this will be helpful as we understand the functioning of a cell. DNA itself, again, has many, many enzymes that are going to enable it to replicate. As we talk about DNA in further detail and cell replication, we'll go into these details as well. So there you have a double helix being split open and making two copied double helices with it. Now that's nucleotides, an overview, but if you want to look at the overview of all of our biomacromolecules, this is a great slide to do that. So I encourage you to go back and review all the information on the biomacromolecules, because we're going to now take those molecules and start to assemble them into cells.